Hello, and thank you for joining us for 3D cell-based models for regenerative medicine and drug discovery, sponsored by AMSBIO and 3D Biomatrix, and presented by Bioscience Technology and Drug Discovery and Development. I'm your moderator, Robert Fee, Editor-in-Chief for Bioscience Technology. Two-dimensional cell cultures provided a number of breakthroughs for understanding human tissues and diseases, as well as for discovering and testing new drugs. However, 2D cell cultures have limitations, such as limited cell-cell and cell matrix contacts, flat cellular morphologies, and a lack of realistic mass transfer gradients. As a result, technologies that facilitate growing cells in three-dimensional configurations have been developed to make cellular behaviors in vitro better resemble the body. Today, two leading experts in the field will explore the potential of 3D cell-based models for regenerative medicine and drug discovery. Before we begin, I'd like to explain how you can be a part of this webinar. A question and answer session follows our final presentation. At any time during the webinar, you can type a question to the panel through the presentation console. We'll answer as many questions as time permits. Okay, now that you know how you can participate, let's get on with the show. Our first speaker today is Dr. Elad Katz, an internationally experienced senior scientist with 10 years experience of work improving cancer research and translating it efficiently into clinical practice. Currently, Dr. Katz focuses on providing a service to develop and implement physiologically relevant cell culture models in oncology and related fields. Prior to joining AMSBIO, Dr. Katz authored more than 20 publications in the fields of developmental biology and cancer research. He also runs a journal club for cancer models and translational medicine, where he shares his passion in improving cancer research and translating it efficiently into clinical practice. Today, Dr. Katz will assess the impact on the physiological relevance of in vitro cell-based models using traditional ECM proteins, Alvatex scaffolds, and low adhesion plates in drug discovery and disease modeling applications. Dr. Katz, thanks for joining us today. I'm looking forward to your presentation.
Thank you, Rob. I will speak today about how to introduce physiological context into the way that we do cell-based assays, especially in drug discovery. Physiologically relevant cell culture is a fairly new concept based on what we typically shorthand as 3D culture. The principles of that concept are three. One, we're really trying to introduce into our in vitro systems the right cellular context. I will concentrate today about the extracellular matrix, but really the entire environment required for regenerative medicine, oncology, or other disease studies needs to be taken into account when we do drug discovery. That will improve the functionality of the models that we are using, and the most important thing, it would really lead to better predictive power in drug development, because attrition rates in most diseases are still appallingly high. Now, we will concentrate today on three different technologies from AMSBIO. The first one would be a new high-throughput assay system, especially designed for oncology, although we suspect that people in other fields may also find it useful. Then we will go for a plastic scaffold solution, concentrating on regenerative medicine and co-culture solutions in a variety of disease areas, especially stem cells. And lastly, I will be expanding from 3D into other ways of doing physiologically relevant cell culture by showing you how we can tailor our cell-based assays using biomimetics. The first technique is what we call 3D spheroid-based assays. This is a new assay system that relies on low attachment plates to generate spheroids composed of cancer cell lines and extracellular matrix. Now, these assays are either for proliferation or invasion, but the most cool thing that you can see here in the picture is that you can actually combine this with other microscopy or imaging-based techniques to really get an awful lot of information right from one plate, 96-well plate. Now, how does this work? This is a simple demonstration of the proliferation assay. You resuspend your cells in what we call spheroid formation matrix and seed the cells in 96-well plates. In each of the plates, on the low adhesion U-bottom, you will get a spheroid form, and within a week or so, you could run a simple colorimetric or fluorometric assay to see how the cell numbers have changed. That doesn't stop you from imaging the spheroids as they grow. I will show you a demonstration of that in a moment, or doing any microscopy-based approach in parallel to the quantification of proliferation. So here is a simple example of that using MDA MB231s. These cells, without the spheroid forming matrix, would not form a proper spheroid, so they are really good to demonstrating how this matrix generates spheroids in the well. And what you can see, if you follow this day by day, the spheroids grow in area. You can quantify it again either by ImageJ or similar software, or by a direct proliferation assay such as MTT. And if you add your favorite drug, just on the right-hand side, you can really quantify the effects on the size of the spheroid very simply. A similar assay is the invasion assay. The first few steps are very similar. You form a spheroid, but now after 3 days, you actually add another special matrix, which we call the invasion matrix. And now your cancer cell line will expand and invade into the matrix. That enables you not only to look at proliferation, but also at the invasive properties of your cells and how specific drugs, or in fact a genetic manipulation, would affect both of these processes at once. Now, here is a simple demonstration of how the invasion would differ between different cell lines. These are very typical cancer cell lines from 4 different kinds of cancer, and what you would notice is that within 4 days or so, post-embedding in the invasion matrix, one can see very different sprouting patterns, and these are really specific to the cell line that is being tested, and could be quantified using, again, ImageJ or any other software that you like, in parallel, again, to any other assay that you would want to run on the 96-well plate. Here is a small demonstration of how would you do that. Again, with MDA 231s that give you this pattern without any drug involvement. If we introduce chemotherapy, you can see that this does affect the cells that are invading locally into the matrix, but in fact, you never really reverse back to the spheroid, which is demonstrated here by no invasion matrix. So you're getting already some sense of what processes might be affected by your drug in parallel to each other, rather than running only a proliferation assay or only invasion assay, as typically is done in cell-based assays. Our second technology that I'd like to talk about today is the Alvatex technology. This is a specially constructed tissue culture plastic. And in this case, we generated well-designed voids where cells could grow into and generate 3-dimensional structures. Now, those voids are reproduced across the plastic in known dimensions, and they have interconnects to let the cells talk to each other. Now, this is available in a variety of formats, but the one that I would like to concentrate on today is the 96-well plate, which you can see on the lower right, because this is a powerful tool for drug discovery. So here, this is how you could do this with oncology. We will move to regenerative medicine in a minute. This is just a typical result obtained using tamoxifen-sensitive cell line called MCF7. It's a breast cancer line. And what you can see is that these cells, after being seeded into Alvatex after 3 days, were exposed to tamoxifen. And there are striking differences between cells that were just on flat 2D plastic and those that were seeded into Alvatex. What you can see is really that there is much more resistance in the cells that are in the Alvatex. That's the red lines after 24-hour exposure or 72 hours exposure. The IC50s are 3 to 4 times bigger. So it's not, you can see in panel C that it's not that the cells are not dying eventually, but you really need much higher doses. And this is something that we know that in the transition between flat plastic to tissue is different. So this was a very good indication that we're really getting in Alvatex structures that are more similar to the cancer that we are trying to study. Now, another example is the HCT116 cells. These are colon cancer cells, and again, they are very sensitive to 5-FU, probably the most commonly prescribed chemotherapy in the world nowadays. And what you can see is that, again, culturing them in Alvatex confers resistance that it's usually not seen in the typical way that we do cell-based assays. Now, if we move to regenerative medicine, I have a few interesting examples for you. If we actually concentrate on the bottom left panel first, what is beautifully demonstrated here is that you could take rat hepatocytes and actually regenerate the basic unit of the liver inside the Alvatex matrix. Now, this is electron microscopy. This is something that you don't do every day to look at it. So what we did is looked at CYP activities and again compared 2D to 3D in Alvatex. And I think that you don't need to be a liver expert to really see that there are very good differences. The activity of those critical liver enzymes is maintained much longer. In fact, we're not much longer than 72 hours in the Alvatex system, something that is impossible to achieve just on plastic. Now, here is another example. This is from bone function. And again, if you see how the bone markers are maintained once you differentiate mesenchymal stem cells into bone inside the Alvatex scaffold, you really see that striking differences. The bone markers are maintained to 2-3 weeks or even more in the Alvatex, while you can hardly get any activity at all in the normal plastic petri dish. Now, just to confirm this, this is a little bit more about the bone. You can see really calcification being done inside the Alvatex, something that, again, is impossible to achieve on flat plastic. So this just demonstrates you in one way how well one could differentiate and functionalize stem cells in a 96-well plate, and therefore really start taking this into the drug discovery level where you don't need to do one sample at a time. Another of my favorite examples is this neural differentiation. And if you concentrate on the right-hand panel, you can really see, I hope, the functional neurons growing so well inside the Alvatex, while the growth and differentiation into the is so much poorer in comparison. Taking that one further into the Coculture system, you can see this fantastic experiment here where you grow neurospheres on top of the Alvatex. And if you compare A and B, you can see that the co-culture of neurospheres and glioma cells, U118, really stops the neurons from growing. You get absolutely beautiful pictures in C and D showing you how poor the neurons are growing in the presence of the glioma cells. And this is by a simple co-culture of generating neurosphere on top of the Alvatex. This is a very flexible system. This is a plastic scaffold, and therefore you can co-culture in many, many ways your 2 and possibly even 3 different kind of cell types in one culture system, which again could be done in a 96-well format. Now, lastly, I just want to expand a little bit that concept of 3D into how we can really design our own environments for cell-based assays. And for that, we use a new tool called Matrix. This is a technology that uses the backbone of a muscle adhesive gene, so it's completely xeno-free. And we introduce a peptide derived from extracellular matrix, in fact, from any protein, into the peptide. This is demonstrated here with natural fibronectin on the bottom left. We can put up to 3 different kinds of peptides, either in one muscle adhesive protein or in more of them. And we can really expose the cells to a known protein, known quantity that is introduced into any system that you like, either in solution or as an adhesive protein. And really, you do not have to worry about batch-to-batch variation. You don't have to worry about the quantities not being quite right or the epitopes not correctly exposed. So you may ask yourself, what is that good for? This is really good for either straightforward replacement of an existing extracellular matrix that you use. This is a fine example of that. You can see that 2 types of neuron cells really differentiate and grow very well on the matrix laminin in this case, just as they would do on the natural matrix protein. One fine example of using matrix biomimetics is really demonstrated here, where UVEC cells were exposed to a big range of heparin binding domains and integrin domains, where on a single 96-well plate, one could look at what these cells adhere to, what they would proliferate to or differentiate to, and move that into the way that you normally do your cell-based assays. Furthermore, this could also be integrated to other 3D technologies, such as the Alvatex, as I mentioned before, or low adhesion plates, or any other technology that you like, now functionalizing your cell environment to the exact purpose that you want to do it. To recap the technologies that I introduced today, I would like to just go quickly through their biggest advantages. The spheroid-based assays are really enabling someone that is doing oncology drug development to detect proliferation and or invasion and or any other cellular processes that they might be interested. We have people already talking about doing high content analysis as well inside those 96-well plates. Alvatex is really powerful in the co-culture arena because you can do different kind of stem cell differentiation in the presence of more than one cell type. Of course, you can, again, introduce any matrix protein that you want into the system, and we have some examples of that. One of the other things that we haven't had much chance to talk about today is really that Alvatex is completely compatible with histological methods. You can get protein extracts from the Alvatex. You can do full genetic analysis of DNA, RNA, or any other tissue culture approach that you're already using in your lab. Now, lastly, the matrix mimetics, again, could really allow you to design specific environments, and we have great interest from people that have for example, cell lines that could simply not grow at all ex vivo, and now they're looking for ways to specify the environment and to develop a whole cell-based assay using these biomimetics to study the function in vitro. I will finish this just by saying something more general about AMSBIO. We really have in-house extensive modeling expertise, so please do contact me if you wanted to ask something specifically after this webinar. We have already strong links to both academia and industry. We work with the major pharma companies, for example. And perhaps the most important thing that I'd like you to take off from this webinar, we do not believe that there is a one-fit-all approach. You have to ask, what is the readout that I'm interested in? What is the cell type that I would like to study for drug discovery or any other purpose? And really follow that rather than saying, this is the one model that I like and therefore I will make my disease work inside this model. So I will finish with a brief showing of the different kind of products that we spoke about. Matrix, I should highlight that there's over 60 of them at the moment. The rest are slightly smaller numbers of products. They're all available to order now. I would like also to highlight that we have other products that I haven't spoken about today in the physiologically relevant cell culture arena. These include basement membrane extracts. Specifically, those that we are excited about at the moment are those that are used by various groups around the world, especially in Europe, to grow organoids ex vivo, to enhance their xenografts. But we also have simpler solutions such as superior low adhesion plates that are becoming very, very popular because we have them in every conceivable tissue culture format. We also will have other technologies coming soon. I could not have the time here today to expand on that, but again, if you have a problem, please contact me and I will try my best to help you tailor our systems to your need. And I will end up with a shameless plug-in for a workshop that I will be running in Dublin next month, which enables you to do this with me. Thank you very much for your time, and I'd be delighted to answer any of your questions.